Good afternoon. Welcome to the webinar Gaucher and Parkinson, the early signs and risks careers. I'm Biljana Jovanović from International Gaucher Alliance. I'm director and vice chair, and I'm also Gaucher patient type 1, and I will be your host today. I will just introduce my colleague Margaret from French organization. Just to put spotlight on Margaret. Hello.
Hi, everyone.
Introduce yourself.
Okay. Well, as Biljana said, my name is Margaret Giuliani, and I'm the IGA representative for the French Lysosomal Association. I was diagnosed with Gaucher disease in 1982 and with Parkinson disease in 2015. Last year, the IGA asked me to lead a project concerning Gaucher disease and comorbidities. So starting with Parkinson's disease, since we know there are others, the aim is to build a community where people concerned can share their experiences, their questions, their issues, and their fears. Because while a lot has been said regarding the link between Gaucher and Parkinson's disease, there's a lot of misunderstanding and fear of the future among Gaucher patients and carriers. GBA-related PD patients would also like to understand more about GBA. So since knowledge is the antidote to fear, I want to thank our illustrious speakers for sharing their knowledge with us today. It's now my great pleasure to pass you over to our first speaker, Professor Ari Zimran. Thank you.
Just a second.
Just before we start, excuse me, I would just say that this webinar will be recorded and it will be possible to look afterwards. We will have all three presentations and then afterwards questions and answers. And also I'm asking Q&A box in the bottom. So, excuse me, Professor Ari.
It's okay. Hello from Jerusalem. I think it's a good evening for many of us in Europe, but maybe nice afternoon elsewhere in the globe. And thank you for the invitation. It is definitely, I would say, the most burning topic today in Gaucher, as well as a growing important topic for the larger community of people who suffer from movement disorders. This is my disclosures, and this is the second more important disclosure that I'm not a neurologist, but I'm working with close collaboration with two movement disorder specialists. One is Dr. David Al-Kadir from Hadassah University Hospital, who is also a visiting professor in our hospital, because until recently, we did not have a movement disorder specialist. We now have Dr. Gilad Yahalom, who used to be a moving sport expert from in Sheba. And luckily, we have Professor Svenningsson from Karolinska Institute, whose role is not just to educate us about Parkinson, but he should correct any mistake that I may say with regard to Parkinson, because my point of view is coming from the world of Gaucher disease. Now, many of you have seen this scale of historical milestone in Gaucher, where the upper scale has to do more with diagnostics and the lower scale more with therapeutics. What I can tell you is that while the first association between Gaucher and Parkinson was reported from our group and collaborators in 1996, it has taken 2 decades before clinical trials were started addressing this specific association. So now I see that I'm... Anyway, so we have 2 clinical trials for Gaucher-related Parkinson. One is an investigator-initiated research led by Professor Tony Shapira from London, and the second is a multi-center, multinational clinical trial that is conducted by Sanofi Genzyme, and is still, I think both are still ongoing, and only recently there were a little bit of early reports. So Parkinson is the second most common neurodegenerative disease, second to Alzheimer, and it is characterized by slowness of movement, what we call bradykinesia, shaking, rigidity, impaired gait, and many other features that probably will be presented by the next speaker. From the pathological side, we are talking about aggregates of alpha-synuclein in the brain, particularly in the midbrain in the area of substantia nigra, something that can be also seen by different imaging modalities. Now, Gaucher is a rare disease. So we are talking here really about an interesting, unfortunately interesting combination between, on the one side, a rare genetic disorder, and on the other side, a very common neurodegenerative disease. In this relation, it's important to know that Gaucher is not just a glycolipid storage disease, but also a protein misfolding disorder. And many of the neurodegenerative diseases have to do with misfolding of proteins. Now, this is important not just as a general understanding of the pathophysiology, but if we understand why there is this link, we can develop specific treatments. So this is something very important. So the 4 topics I will try to cover relatively quickly in the next less than 20 minutes by now is there are 4 key features. One is the realization of the fact. The fact is that both patients and carriers of Gaucher disease have a higher risk to develop Parkinson. If you like, we can already tell you that Gaucher abnormalities in the gene that encode the glucocerebrosidase is the #1 genetic risk factor to develop Parkinson. There are also many genetic and non-genetic diseases, risks. I want to introduce you the concept of prodromal Parkinson. These are signs and symptoms that precede the diagnosis of the motoric Parkinson disease that all of us know. And this is also very important because when we talk about a population at risk, like the GBA carriers and Gaucher patients, we may be able to detect those who have more than the just statistical risk, whether it's a 3-fold, 5-fold, etc. So we'll talk a little bit about prodromal PD. We'll return to briefly review the 2 main schools, I would say, that explain the underlying relationship between the genetic abnormality and the Parkinson. And we'll briefly tell you about different investigational therapies. Some of them are just in the potential, but some of them are already in either preclinical or clinical development. So overall, there are the bad news and the good news. The bad news are that there is a higher risk. If we talk about 1% of the population over the age of 60 has a risk to develop Parkinson, we talk here about 3% to 5% for the patients, and it may be up to 15%, 15-fold extra risk for carriers of bad mutations. The good news is that the majority of the patients and the carriers do not develop Parkinson. The second bad news is that when Parkinson is developed in a background of a GBA mutation, whether a Gaucher or a carrier, the clinical course is more severe than of the so-called idiopathic Parkinson. But again, for the good news is that once we understand the relationship, we may be able not just to treat, but also in the future to prevent this added risk. So as I told you, and I don't have enough time to tell you, but actually this first paper that was very hard to publish, it was rejected several times until it was published in Quarterly Journal of Medicine. It is a mini-series of 6 patients with Gaucher who developed, we called it Parkinsonism because some of them did not respond to L-DOPA, which is maybe part of the definition, but the commonality between the 6 cases was that they all had relatively mild Gaucher disease and severe Parkinson. And the fact that they had an earlier age of onset of the Parkinson is a reflection of the severity. Shortly after this report, Ellen Sidransky diagnosed the first patient, and she at the NIH decided to dedicate her career to the research of these associations. So we went silent for a few years, but by now there is a huge, it's a textbook knowledge by now. Now, the world of Gaucher, I don't know how it works in movement disorders, but Gaucher disease is full with controversies, sometimes even unfriendly controversies. So the claim that patients with milder Gaucher disease tend to have a higher risk was not accepted by everyone. This was our first observation. Secondly, it was supported from data from the ICGG registry, where they found that the milder Gaucher phenotype occurred in a majority of those who developed Parkinson. At that time, there were 68 Gaucher patients who developed Parkinson. Our colleagues from the US, led by Professor Pramistry, had this opinion that it is not related to disease severity, and also they disagree with the magnitude of the problem. If we talked about 3 to 5%, they meant something over 20-fold lifetime risk to develop Parkinson. There was another disagreement, is that I had this feeling that perhaps paradoxically, enzyme replacement therapy was facilitating the development of Parkinson in patients with Gaucher, and they claim that it was the other way around. They say that, and we see here a citation from the late Henry Termere, who was the legendary president of Genzyme. As Gaucher patients began to live longer, the doctors started noticing that they had a greater than normal incidence of Parkinson. So I disagree, because I do not think that there is a significant shorter life expectancy to patients with Gaucher. And the Henry Mencken, who was a legendary orthoped surgeon in the time where he used to see only patients with severe disease, he used to tell the mothers who came with children who suffer from bone problems, I have a good news, bad news. The good news, your child will live just like his unaffected siblings or anybody else. The bad news, that they will suffer all their life from bone pains. So enzyme replacement therapy has removed the majority of the skeletal problems when the diagnosis is made on time and treatment is starting earlier, but it might have removed some protective factor that exists to the patients. And I did my training in 86 to 89 under the mentorship of Ernie Beutler. At that time, there were few Gaucher centers in California, in New York, in Washington. Nobody has a patient with Gaucher and Parkinson. Today, everywhere you go, every group of 20 patients will have one with Parkinson. The second interesting aspect is that the risk is not just for patients, but also for carriers. And again, I don't have time to discuss how things have evolved, but it is today, again, a textbook knowledge. And we have enough studies, meta-analysis and other, that document that the risk is greater for carriers of bad mutations. You know, bad mutations are those that can be associated also with neuronopathic Gaucher disease, and the risk is smaller for patients with mild mutations. In fact, some of the mutations in the glucocerebrosidase genes are not even a disease-causing alleles. And again, I don't have time to talk about it, but you do need to trust me on this one. Ernie Beutler used to define the severity of the mutation based on the ability to lead to neuronopathic Gaucher disease or not. So N370S, a prototype of mild mutation, will always be associated with type 1, even if the other allele is severe. severe and severe renal will lead to neuropathic Gaucher disease. In the Parkinson literature, you can talk about N370S versus non-N370S. And importantly, it's the same mutated gene that is affiliated both with Parkinson and with Gaucher. Importantly, with neuropathic Gaucher disease, and again, I'll come back to this. And this is just one of several studies that document the more severe clinical course of individuals with mutations than those without mutations. So here we see all GBA carriers, and you see that the life expectancy and the progression of the disease is worse than the non-GBA carriers. And within the carriers, those who have bad mutations are doing worse than those who have milder mutations. So this is just an anecdote to show how Gaucher moved from being a recessive disease, Gaucher disease, and the Parkinson part is dominant because it's enough to have one affected parent to pass on the risk. But the penetrance is not as big. So we are facing this paradox that while the patient with Gaucher who develop Parkinson have typically a milder phenotype, milder mutations, the carrier of the more severe mutation have a greater risk. So how can we understand it? And this is quite clear to me. Maybe the explanation is incorrect, but the observation is crystal clear. The difference between Gaucher patient and Gaucher carrier is the storage. Carriers don't have storage, don't have Gaucher cells. Patients have Gaucher cells. The fact that the prevalence of Parkinson is equal between Gaucher patients and carrier would not make sense when you have twice the amount of genetic defect or misfolding proteins. So if we look at this Gaucher cell, if this cell, we already know that it's not just a silent, large macrophage, it also secretes a lot of cytokines and other substances. It makes sense that one of the substances that is excreted by the activated macrophage, Gaucher cell, is neuroprotective. And this is why we didn't see patients with Gaucher and Parkinson before the advent of enzyme therapy. As enzyme therapy is available, and the higher the dose, the greater the risk, the longer the treatment, the greater the risk. When these macrophages are not secreting anything protective anymore, then there is a greater risk for the Parkinson. Now, a few words about the prodromal Parkinson. Basically, non-motor symptoms like reduction in smell, sleep abnormalities, depression, constipation, and several other findings occur around 10 to 20 years before the diagnosis of the typical Parkinson is made. Now, in the past, nobody cared about so much about it because there was nothing to do anyway. And when the patient was first diagnosed by the neurologist, many times he was asked about the smell. Usually it's a male, and the wife would say, oh, he doesn't smell my food for so many years. Now, if we know that we can detect earlier the risk, and once we have something to prevent it, maybe we will be able to either delay the typical manifestation of Parkinson, or hopefully even maybe to prevent them. But we are not there yet, but this is definitely a direction of active research, and some of the leading centers are indeed the one that I mentioned before by Professor Tony Shapira in London, Ellen Sidranski and the NIH and others. Now with regard to the, I told you, Gaucher always have arguments about mechanisms of very hot debate. Are we talking about gain of function or loss of function? What does it mean? A gain of function means that when you have a mutation, it changes the activity of the protein, so the protein does something else. That can be something bad in this case. Or loss of function, which is more common. Those are mutations that lead to abolish of protein function. So in the case of glucose residues, mutation may lead to lack of active degradation of the glycolipid, so storage of glucose cerebroside. And this is not a theoretical issue. This really leads to different therapeutic approaches. Because if we believe in the loss of function, we will give SRT, we'll try to reduce the content of the glucose cerebroside in the dopaminergic neurons, or we'll try to give enzyme replacement via opening the blood-brain barrier this way or another. If we believe more in gain of function, then we want to improve the folding and to cut this link between the mutant protein and the alpha-synuclein, and we are talking about pharmacological chaperone and related compounds. Of course, in medicine, many times, it's not just one mechanism that plays a role, and it could well be that both gain in function and loss of function are taking place, and both need to be addressed. Now, again, for the sake of time, and also the patient really don't need to see every single detail, but for the doctors among you, I will tell you that the loss of function is mainly based on animal models or induced cells. And the mouse model that has led to the development of venglustat currently in clinical trials is a model of a Gaucher mouse with 2 mutations, homozygosity D409V, that does not exist in human people. And then there is also mutant mouse model for the alpha-synuclein. So recently, a group of doctors convened somewhere in Europe, and again, this paper was led by Ellen Sidransky. I like her title, Substrate Reduction Therapy for GBA-associated Parkinsonism. Are we betting on the wrong mouse? This was published as Viewpoint a few months ago. I mean, time flies. Last year in November, and you are welcome to read it. In support of gain of function is the observation that carriers and patients have roughly the same risk of Parkinson. There is no solid evidence that carriers of Gaucher have any substrate accumulation, and 50% of the wild-type enzymatic activity has been always believed to adequately cleave the glycolipid. And so the idea of excess glucose cerebroside may not exist. Again, lack of correlation between Gaucher severity and risk of Parkinson also speak against the storage. And there is a chart I show you here. This is not published yet, but it was presented in a couple of posters. Dr. Al-Qadir measured with the help of the Centogene team the lyso-GB1 levels, which is the glucose sphingosine that reflects the amount of the storage of Gaucher cell burden. And you can see these are all untreated patients. Gaucher, no ERT in blue, and Gaucher, no ERT, they develop Parkinson in red. And you see that the red spots are really showing the lowest level of the lyso-GB1. By now, we have 2 more cases that confirm the same observations. And together with the other animal models, remember I told you we were betting on maybe the wrong mice, so there is another mice and another fruit fly models that support the gain of function hypothesis. And Mia Horowitz, for example, demonstrated in nice Parkinson-like models of Drosophila Parkinson that was derived from overexpression of mutant glucocerebrosidase, that when you treat them with ambroxol, a pharmacological chaperone, there was improvement in the function. They could fly again, they live longer, and they suffered from less inflammatory changes. And Tony Shapira in London had a similar observation in animals, and because of that, he also conducted this IIR with ambroxol. So there are 2 ongoing clinical trials according to the 2 different approaches, and both of these studies have recently demonstrated that both are going in the right direction with regard to impact on biomarkers, but it's too early to see what will be the impact on the actual neurological features of Parkinson. So very quickly, because I'm running short in time, what do we have now in addition to Venglustat, the SRT that is given to many GBA carriers, and in addition to the Ambroxol that is given only in one clinical trial or two, but it has been, because this is an over-the-counter cough medicine, many patients can buy it. They just need a higher dose than the dose used for cough. And many patients with Parkinson who are GBA carriers are taking it off-label. But formal clinical trials are done for gene therapy. And you know, gene therapy has two basic types. Again, either direct delivery, what we call in vivo gene therapy, where you inject the engineered viruses directly to the brain. And this is the case of Prevail Therapeutics in New York. They are conducting an international study, phase 1-2, to treat GBA carriers or Gaucher patients with their gene therapy. This is going to deliver the particles to the brain that will produce glucocerebrosidase, so correction of the loss of function. There is another interesting company that I just met recently, but there are several ones, where they are trying to open the blood-brain barrier. You know, this is the protection the brain has from all kinds of bad things. But on the other hand, like enzyme therapy does not impact on the brain because it's a big molecule that doesn't cross the blood-brain barrier. And with a very sophisticated technology of focused ultrasonography, proudly I can tell you that this is an Israeli invention from about more than 10 years ago. They already have an FDA protocol approved for people with essential tremor. They've studied for Parkinson. And they are looking into also treatment of neuronopathic, not just Gaucher, but neuronopathic LSD by selective and transient opening of the blood-brain barrier. Just to show you how many things are in development. Many of you are familiar with Orphazyme, the Danish company that developed arimoclomol for Gaucher disease, all types, and for GBA-related Parkinson. If you look at the website, the GCA's Parkinson project is preclinical, but it's about to begin soon. Arimoclomol is an interesting small molecule that amplifies the activity of the heat shock protein, mainly HSP70, that it is in contrast to Ambroxol, which is a pharmacological chaperone. You take it from the outside and bring it to the cell to improve folding the protein. There are natural occurring chaperones, and one of them are those compounds, and by improving their activity, we hope to achieve correction of the misfolding of the glucocerebrosidase, thereby prevention of the later aggregation of the alpha-synuclein. So my dream is to use the information we had about prodromal symptoms to screen GBA carriers and Gaucher patients for these signs and symptoms. That what is interesting is that each one of them can be measured both qualitative and quantitative. And I think that once we find the carrier that has a significant number of prodromal features, he or she can be a candidate for a placebo-controlled trial that will test, let's say, Ambroxol plus something, arimoclomol maybe, and see if there is a difference between placebo and the investigational therapy. If I can convince you guys, we see what the neurologists are going to say, or the regulatory, that if I correct the smell sensation or some finding, you know, we measure alpha-synuclein in red blood cells, we do ultrasonography of the brain. If we correct it, then maybe we can prevent it. Now, why I believe we can correct it? Because just as we didn't know that there is such a significant concept of prodromal features, we used to say that 70% of the dopaminergic neurons need to die before Parkinson emerges. But as I told you, it's the same misfolded mutant glucocerebrosidase gene that in other neurons is leading to type 3 Gaucher disease. And when patient in Japan with severe myoclonic epilepsy, ataxia, and other findings started with Amboxol, high dose, within weeks, they got better in parameters that heretofore were considered to be irreversible. So if you could, maybe of those 10%, 20%, 30% dopaminergic neurons that you kill them in the lab, maybe in reality they are salvageable. So I think I'm a believer. Unfortunately, it's not very easy to raise funds, but we have a guy from J. Fox Foundation. People were not so enthusiastic about prevention. So I will not repeat all the conclusions because it was a short talk, and you remember. I just want to mention that our unit is inviting GBA carriers and family members over the age of 40 to join this study. And this is my group here. In yellow star is my successor, Professor Shoshana Revel-Vilk. Professor Mia Horowitz taught me about misfolding proteins maybe 15 years ago. And Professor Rolfs is helping us with biomarkers. I mentioned before lyso-Gb1, but there are other markers as well. And I thank you for your attention.
Thank you very much, Professor Ari. Now, Professor Per Svenningsson from Karolinska Institute. Thank you for joining.
So sorry, I need to unmute that now. Now it should be fine. Okay. So thank you very much for your very kind invitation. I'm very honored to attend this webinar, and I was very impressed of the lecture that we just heard from the Professor Zimran, who is really a legend in the field. So there will be a little bit of overlap between our lectures, but I start off a little bit more from the Parkinson side. So hopefully I can add some information to what Professor Zimran just told you. So here are my disclosures. So there are more than 7 million people in the world that have Parkinson's disease, and there are more and more people getting this disorder because the strongest risk factor to develop Parkinson's disease is an old age. So we will have the double amount of Parkinson's disease patients in 2030. Parkinson's disease is diagnosed based on movement disorders. So there is the slowness of movement or bradykinesia, rigidity and tremor. And patients also often have problems with the speech and their posture. I wanted to show to you some video films showing these characteristic symptoms of a Parkinson patient. So this is then patients who have had the disorder for some years. And I start with the bradykinesia, or the slowness of movement. So this man walks towards us, and you can see that he has poor movement repertoire. He doesn't swing his arms so much. Tends to be a little bit bent towards one side, and we call that the Pisa phenomenon. He walks with a shuffling gait and short steps, and you will see now that when he tries to turn, that he freezes a little bit into the ground. Patients often have problems to initiate walking, but once they start to walk, they can walk rather well. And some persons with Parkinson's disease find it's actually easier to run than to walk. So this is what we call the bradykinesia, or the slowness of movements. On the next film, I will show the rigidity, and that is something that we often find in the upper limbs. And, oops, sorry, no, I have to go back. So this is the way that we examine persons with Parkinson's disease, and this is then an individual who don't have Parkinson's disease. There is a smooth movement here in the elbow. If we then examine a patient with Parkinson's disease, they have what we call a cog wheel phenomena. And this goes in both directions when we do these movements. And this is what we then define as rigidity. And it's very characteristic for Parkinson's disease. And it's mainly in the upper limbs, a little bit less so in the lower limbs. The third characteristic symptoms that about 60% of the Parkinson patients suffer from is tremor. And I will now show you a film with two ladies drinking tea and coffee. And one has what we call the Parkinsonian rest tremor, and the other woman has another form of tremor that is very common that we call essential tremor, an action-based tremor. So here you will see now the two ladies. So this lady has the rest tremor. So this is something that happens when you are not doing any movements. And it's a little bit more in the right hand than in the left. But you will see now that when she drinks her tea that it disappears. So the Parkinsonian tremor disappears upon movement. That's very characteristic. So she had no problem at all to drink her tea. But now that she has done that, it reappears, the tremor. So it is at rest. Whereas the other lady here who drinks coffee has her tremor upon action or upon movement. This is called an essential tremor. And the treatment for essential tremor is very different from Parkinsonian tremor. So there is no overlap in these pathologies. And you can see that she has her tremor when she moves. And very soon you will see the two different forms of tremor at the same time here. The rest tremor and the essential action tremor. There is about, like I said, 60 to 65% of persons with Parkinson's disease that have tremor. We also have a type of Parkinson's disease that we call akinetic rigid Parkinson, where you don't have the tremor. So there are two major pathological hallmarks of Parkinson's disease. There is the loss of the nigrostriatal dopamine neurons. And you can see that on this slide that in the brain section from the Parkinson's disease patients that there are fewer of these dark neuromelanin containing neurons, especially in cell bodies that project to a region called putamen. And this is also something that we can see in the living patients and that we often do when we put the diagnosis of Parkinson's disease. We do that with different imaging methods. In this slide, I show images from a DAT PET study. So it's radioligand that binds to the dopamine neurons. And you can see the dopamine neurons, should try to point here perhaps, here at what we call the terminal region, at the endings of the dopamine neurons, but also at what we call the cell bodies, where this region where we have this dark neuromelanin containing cell bodies. And a little bit related to what Professor Zimran mentioned, it's very interesting in the beginning of Parkinson's disease, at earlier stages, we have a very clear-cut loss of the terminals of this dopamine system. So the pathology of Parkinson's disease seems to happen in the terminals of the neurons, whereas at the cell body level, the dopamine signal is still present. So we do think that if you can find a therapy that could slow down the progression of Parkinson's disease, that there is a substantial amount of dopamine neurons that are still, where you still have a cell body, but where the terminal may be dysfunctional. So we think that there is a quite large percentage of cells that we can help to survive if we find the proper therapy. However, over time, we also lose the signal of the dopamine neurons in the cell body and in the terminal region. However, this dopamine deficit that we see in Parkinson's disease is something that we can treat. And the major treatment still for Parkinson's disease is a replacement treatment or restoration treatment with the compound called levodopa. And since I'm Swedish, I'm proud all the time to say that it was a Swedish pharmacologist, Ari Karlsson, who first found this. He did experiments as illustrated on this slide in rabbits, and he gave the rabbits a pharmacological agent that depleted dopamine and also other so-called monoamines. But he found that when he gave the rabbits levodopa, which is the precursor of dopamine, that the rabbits got back a very good locomotion and movement repertoire. And he published this now more than 60 years ago. And he could also show that levodopa or L-dopa was the precursor of dopamine. So by giving levodopa, we can restore a lot of dopamine function also in Parkinson's disease. And I show this slide because it's a slide illustrating that Ari Karlsson, based on his work, was awarded a Nobel Prize in Physiology and Medicine in 2000. And this is a collage actually by me showing a Nobel Prize ceremony. And I have, like I said, it's collage. I didn't find the real image of this ceremony, so I have pasted Ari Karlsson, a little bit larger version also of Ari Karlsson, into another ceremony from 2013. But anyway, so Ari Karlsson was awarded a Nobel Prize in Physiology and Medicine for his discovery on the dopamine transmission. And many people have then followed the work of Ari Karlsson and developed additional therapies, most of them based on the dopamine system and to restore the dopamine transmission in Parkinson. So at the moment we have up to, I would say, 15 different therapies where we can help and treat our Parkinson patients. We combine different ways of strengthening the dopamine system. And we also have another treatment that is very good for many persons with Parkinson's disease. We can do something called deep brain stimulation, which is a neurosurgical approach, which is also very good to restore locomotion. However, all these therapies are symptomatic, so they take away only take away the symptoms of Parkinson's disease, and it's of course very important for the quality of the life of the patients, and we really try to optimize the therapies, but they do not slow down the disease progression. So unfortunately, our patients get worse from year to year, although we try then to combine as good as we can all these different treatment options. And when it comes to the real root of the cause of Parkinson's disease, we think that the second pathological hallmark may be more important, namely that the Parkinson patients have something called Lewy bodies, which is aggregated proteins, and the main component in these aggregates is a protein called alpha-synuclein. And we think that this is driving the disease. And there's a lot of focus in the Parkinson field to try to intervene with this alpha-synuclein. So we know that this alpha-synuclein exists at what we call monomers, and these monomers then tend to form larger species that then get insoluble fibrils, and you get the Lewy body. So different pharmaceutical companies try to intervene with this process. There is also ways of trying to reduce the spread of this alpha-synuclein or Lewy bodies from cell to cell. But at the moment, we don't have any therapy. But this is something where there is a lot of interest, and hopefully there can be a development. Anyway, this alpha-synuclein has been shown by a German pathologist called Heiko Brack to spread in the brains of people with Parkinson's disease. And initially, the alpha-synuclein is found mainly in the brainstem, and then it's spreading to engage the dopamine neurons, and then it engages most of the brain. And this spreading of alpha-synuclein or Lewy bodies is correlated nicely with different clinical symptoms of Parkinson's disease. And as Professor Zimran very nicely mentioned, introduced to you, we have what we call a pre-motor or preclinical phase of Parkinson's disease, where people have loss of smell, RBD, which is a sleep disorder. And this sleep disorder is very peculiar. So during the REM sleep, when we dream, we usually, who don't have this RBD, we lose the tone of the body. But if you have this RBD, instead of lying paralyzed and sleep and dream, you start to move. You start to move and you start to scream, and so you live out your dreams. And this can be very disturbing, especially for the partner, but you can also hurt yourself. You can fall out of the bed and so on. So this is something that makes people come to the hospital and get the diagnosis RBD. And we know that if you have this RBD, you have actually a 20-fold increase of developing Parkinson's disease. But also there are other preclinical or prodromal symptoms: constipation, anxiety and stress related disorders, and depression. And we know that if you have a depression, you have a twofold increased risk of getting Parkinson's disease. Now, depression is more common in the society than Parkinson, but this is early signs that the Lewy body's pathology is starting to engage the brainstem. And then when it engages the dopamine neurons, the people develop the movement disorder, the bradykinesia, the rigidity, and the tremor. And then the Lewy bodies continue to engage larger part of cortex and many regions of the brains, and patients develop problems with balance and fall, and they can get problems with the speech. A lot of patients get cognitive decline and dementia, psychosis, and also a lot of problems often with the blood pressure, especially when you have when you stand up, you have something called the orthostatic hypotension. I should say that, I mean, it's not all persons with Parkinson's disease that get all these symptoms, but on average, these are symptoms or signs that the patients develop. It was shown by Ellen Sidransky and colleagues that in these Lewy bodies, you can also find mutated glucocerebrosidase then encoded by the GBA. So now I turn to GBA and Parkinson, and I start with I think, classic slide from the work of Professor Zimran and at his very big Gaucher clinics, where he together with Roy Alcalay studied when people with Gaucher disease or gene carriers of GBA or non-mutated people get Parkinson's disease. And as you can see, the Gaucher patients get Parkinson's disease earlier than both the gene carrier and the persons without any mutations. The gene carriers are in between the persons without mutations and the Gaucher disease patients. And it's about 5 to 10-fold increased risk of getting Parkinson's disease in the GBA carriers. One thing that I think that this slide also very nicely illustrates is that the large majority of both Gaucher patients and GBA carriers do not develop Parkinson's disease. So there is some additional factors. And now we heard this interesting talk from Ari Zimran, where he implies that the Gaucher cells may actually contain neuroprotective factors, and there must be a second hit that makes some people that are GBA carriers or Gaucher patients to develop Parkinson's disease, because the large majority do not develop Parkinson's disease. And then we have this very classical work by Ellen Sidransky, where she looked at how many of the Parkinson's disease patients at different centers all over the world have GBA mutations compared to people without Parkinson's disease. And in most centers of the world, there are a much higher prevalence of GBA mutations in Parkinson patients compared to controls. And especially you see the focus on N370S and the L444P. However, what caught our interest in Sweden is that our neighbors, Norway, that were part of this study, did not really seem to have an increased odds ratio among the Parkinson patients compared to controls. So we have been very then interested to study how is the situation in our country. But unlike the Norwegians, we are a little bit more like the rest of the world. So we have an increased risk of GBA mutations, or we have among our Parkinson patients, we have many more with GBA mutations than compared to the controls, and especially the N370S and the L444P. We have also found an increased frequency of the E326K mutations. This is a relatively prevalent mutation and not so very often found among Gaucher disease patients, but it has been found in several different populations to have a higher odds ratio in Parkinson's disease as compared to normal patients without Parkinson's disease. We have also in Sweden actually found some unique mutations, S107L, that we are now studying in larger sets of samples. If we put the GBA mutation in the context of other genetic risk factors for Parkinson's disease, it is, like Professor Zimran said, the most common genetic risk factor for Parkinson's disease. The risk is it's called a medium risk gene compared to some other very rare genes that we know cause Parkinson's disease, but are much less, are much rarer. And then we have some also other more common genes, but with a lower risk. So it is in the center of the genetic map of Parkinson's disease. And this slide was actually shown by Professor Zimran and illustrates one other aspect of GBA PD compared to idiopathic PD, namely it's the GBA PD has an earlier onset, but also an accelerated cognitive decline. And this is particularly true for the neuronopathic mutations compared to the milder mutations. And that's also shown in another study here from Italy, where they looked at many different signs and symptoms of Parkinson's disease and found that and compared the GBA PD to the non-mutated PD. And again, the cognitive impairment is something which is more prevalent if you have GBA PD compared to idiopathic or PD patients without mutations. But there are also some other symptoms that are more common, like hallucinations. But then I should tell you that hallucination is something that is very, very often occurring together with cognitive impairments. So there is a very strong correlation. So they very often go together. And also that's also true for delusions. But there are other symptoms like anxiety, non-motor fluctuations, these blood pressure problems and so on that are more prevalent among GBA PD as compared to non-mutated PD. So overall, especially for the neuronopathic forms of GBA, it seems unfortunately that the symptoms that a person gets is somewhat more severe than if you don't have a GBA mutation. Now I will just talk a little bit also again about this preclinical phase, or prodromal phase of Parkinson, where we have loss of smell, this sleep disorder, RBD, constipation, anxiety, depression. Because Professor Shapira, that was mentioned by Ari Zimran, has followed patients with Gaucher disease, GBA carriers or control, and studied this type of symptoms. So these are then people who do not have Parkinson's disease, but he studies these prodromal symptoms, and he has found that there is no difference when it comes to smell between, there is no difference, at least when you follow up after 2 years, between the Gaucher patients, the carriers, and the controls. The smell overall may be somewhat lower to start with, but it didn't change over 2 years. There is no change in the cognitive aspects, in cognitive function. Actually, Gaucher patients and GBA carriers and the controls did better after 2 years than baseline. At least with this test of cognition. With another test of cognition, there was some worsening, but this is what we usually use in Parkinson. But what they did find when they followed Gaucher patients, GBA carriers, and control is that this, after 2 years, more carriers and Gaucher patients have this sleep disorder, RBD. They also found that some carriers develop depression. So these are two then prodromal symptoms of Parkinson. And also for the Parkinsonian scale, there was some overall worsening. This is a combination of many different questions, and there is some worsening between Gaucher and carriers. Now, they have also done in the same group another follow-up of Gaucher patients without Parkinson, GBA carriers, and healthy controls. And again, the smell is not that changed. However, this REM sleep behavioral disorder continues to be something that is getting worse in the GBA carriers and the Gaucher patients. The cognition here is not changed. The depression is changed, but it's changed in all groups. So I would say the symptom that is really standing out among carriers and Gaucher patients as a prodromal symptom is this REM sleep behavioral disorder. And I should also mention that after 6 years of following these carriers and Gaucher patients, it was only one person that developed Parkinson's disease. Now, other people have looked at persons who have this RBD, this sleep disorder, and studied, because we know that they have actually a 20-fold increased risk of developing Parkinson's disease, whether it matters if you have a GBA mutation or not. And it seems that the persons who have a GBA mutation, and this RBD, they develop Parkinson's disease somewhat faster than persons without the GBA mutation. And again, the severe mutations are a little more aggressive than the mild mutations. Finally, I'd like to just briefly mention also something about the therapies. So, and this was very nicely introduced and discussed by Professor Zimran. And I come back to that there are a lot of activity actually in this area and different studies that are ongoing. One is this substrate reduction therapy, where an inhibitor of the glucoceramide synthetase is given to patients with that are GBA carriers and have Parkinson's disease. And a phase 1 study has been conducted and finished, and venglustat is safe and tolerable. And a phase 2 study is now very soon finished, and the efficacy results will be released next year. Then there is also 2 studies where compounds that either acts to help the misfolded glucocerebrosidase to go properly to the lysosome, or to directly activate the glucocerebrosidase is tested. So one is the Ambroxol study that Professor Zimran introduced. It's a compound that is actually already used as a cough suppressant and a mucolytic drug. And here again, a phase 2 study. It's a study without any placebo control has been conducted, and it's a safe and well tolerated compound overall. And it had some positive activity on the glucocerebrosidase enzyme activity. And then there is another study that was done at least going on in the Netherlands, where there is an activator of glucocerebrosidase that is being tested. And from this study, it has been reported that it's the compound goes into the brain and it seems not to have any safety concerns. And the last study that I indicate here is the gene therapy, where the glucocerebrosidase gene is delivered directly into the brain or into the cerebrospinal fluid that then and thereby transported into the brain is ongoing. And this, there, to my knowledge, we don't have any results yet, but it's a study that has been started. So to summarize my lecture, there is a 5-fold increased risk of Parkinson's disease in GBA mutation carriers, but it's very important to remember that there's still a low penetrance to develop Parkinson's disease in GBA mutation carriers. The age of onset is earlier and the development of cognitive decline is worse in GBA Parkinson patients than idiopathic Parkinson patients. But what is very good and what was also emphasized by Professor Zimran is that there are several specific trials for GBA PD that are ongoing or that are upcoming. And I would like to acknowledge the co-workers here in Stockholm that work with me on Gaucher's disease and GBA and Parkinson's disease. Thank you very much for your attention.
Thank you so much, Professor Svenningsson. Now we have a presentation from Dr. Marco Baptista. Welcome.
Hi, thank you. I'm just going to share my screen. Great, hopefully everyone can see that. So I want to thank the organizers for having me. It's quite a privilege to be on a panel with such distinguished guests. So quite an honor, and I thank you. Instead of really focusing on a lot of material, which I think has been covered brilliantly by the other panelists, I think I should maybe just address everyone with a message of hope and empowerment and what the Michael J. Fox Foundation can do to help empower everybody so that we can hopefully get to a cure. I have a slide here about what Parkinson's disease is, but I'm not going to go over that since I think it was covered very well by the panelists. I do want to stress, though, that the non-motor symptoms have always been historically something that's been ignored, and I think more and more it's important to focus on the non-motor symptoms because I think it's giving us a better idea also of the pathophysiology of Parkinson's disease and where it might be originating, and also because a lot of these symptoms seem to have a link also with Gaucher disease. So the mission at the Michael J. Fox Foundation is one that is extremely focused. We want to find a cure for Parkinson's disease. I'm not an expert in Gaucher, but I do really realize that a key thing in trying to help patients is to understand the cause of a disease. And in Parkinson's disease, we really do not know the cause. We have some pretty good clues. Some of them are environmental, others are genetic. And so those are the ones that we really want to try to follow and really try to better understand that link. So that's sort of my connection with the area of Gaucher and of GBA. As you can see in our statistics there on the bottom, we were founded in 2000 and we've made a huge effort in trying to get money out to the best research and the best science that's out there. And we're now approaching closely to $1 billion in funded research to date. So we're very proud of that and very excited. In general, at the Fox Foundation, we sort of look at the way that that there's a roadmap to try to find a cure for Parkinson's and to better understand GBA Parkinson's through sort of these different areas of understanding biology, which we call defining, the biomarkers, what we're trying to measure, the therapies, how we're approaching to try to treat patients, and then the tools which help enable this whole process from happening. And I believe Professor Zimran had mentioned just how slow drug discovery is. And that's something that we think about a lot at the Michael J. Fox Foundation, is how can we speed up a very slow drug discovery process. If you go from the years in which genetic mutations have been found to be linked to a particular disease, and then to the point in which there are actual clinical trials. This can span 10, 15, 20 years. So we're trying to do our best to try to speed that up. So here's a snapshot of the portfolio that we have at the Fox Foundation around GBA specifically, and we have similar portfolios around other genetic causes of Parkinson's disease, such as LRRK2. We also investigate environmental causes that may be causing the disease. And here you can see, and I think it's really reflective of the conversations that have been happening here with the panelists, that a lot of our focus is around trying to understand the biology. Why is it that people with GBA mutations or have Gaucher don't necessarily then develop and have the disease? They might be at risk, but majority of GBA carriers and Gaucher patients do not develop Parkinson's disease. So there must be something else that is part of the story that we're not fully understanding. Maybe something in the environment, maybe other genes, and as again Professor Zimran was mentioning, potentially things that are protective in some individuals at the genetic level that is preventing them from converting and getting Parkinson's disease. So we have a lot of projects around understanding biology, understanding the genetics. We also have a lot of effort around biomarkers. How are we going to measure in the clinic? What are we going to measure to best pick the right individuals for the right clinical trial? And I think it's very important that potentially there could be very promising drugs that can be tested, but if they're not tested in the right population, you might not be able to get the, you might miss an answer. You might miss a cure that might be out there. So better ways in being able to measure things within GBA Parkinson's at the preclinical level, and then try to translate that into the clinic. So we also have a section here around therapies, and both of the panelists gave very nice explanation, very nice summaries of the different trials and the different approaches that are out there right now in the clinic. And we find that very exciting. I think this is a very exciting time if you look at the different clinical trials that are there. However, we also know that there's very high attrition in the clinic, and there's a lot of things that fail. In fact, within the CNS field, it's approximately like 90% of trials fail, and their drug never gets to make it to the market. So at the foundation, we're also always trying to see what else is there that we could potentially support. High-risk approaches maybe that not all funding agencies would support, so that we can get that pipeline filled up. So if something drops out in the clinic, we're already trying to move something a little bit more forward. So for example, we're working with different investigators to do more drug screens and try to increase the speed of doing those drug screens against the GBA Parkinson's. We're also trying to better understand the better delivery and better distribution of a drug so that it's getting all to the right areas of the brain. And then finally, we think that tools is crucial to sort of enable all of this. And there's a lot of tools that researchers need. And within the GBA PD area, there are different animal models that are needed. There are different types of reagents, such as antibodies, that are really crucial in facilitating research that hopefully will then translate into things that are going to be promising into the clinic. And finally, I just wanted to end on a note of empowerment. And I think there's been a cultural change over the years where we don't see now that it's simply going to be researchers who are going to solve the problem and deliver the drug for patients. There needs to be a lot of patient involvement, and there are many different ways to do this. At the Michael J. Fox Foundation, we have one of our largest investments is on an observational trial that we call PPMI, or Parkinson's Progression Markers Initiative. And it's just a very large observatory study. There's no drug that's being delivered. And what we're doing is we're trying to best capture a phenotype, a signature of individuals across time, either those that are healthy that don't have any disease, we welcome that participation. We welcome the participation of individuals who have Parkinson's, of carriers of GBA and other genetic causes of the disease, even if they're not manifesting the disease itself, all with the goal of really trying to understand what is it that's happening at the genetic level, at the cellular level, that is causing some individuals to get Parkinson's disease and others to not. So we've already achieved about 1,100 active participants across 33 sites around the world. Our goal right now is to expand that so that we can get potentially 4,000 participants at 50 sites worldwide. So most of the sites are in the United States, but we also have sites in Israel and Canada, in the UK, in Greece, in Austria, in Germany, in France, and I believe I'm missing some others. I welcome everybody to go to our website and see the opportunities that you can have in participating in research. And there's different levels of participation also, that they don't all have to be so cumbersome and so invasive. There's also ways in which you can simply go online, and right now we're living in a world that's very remote. We have an initiative called Fox Insight, where you can just be taking a survey and collecting what we think are very important information from the patient perspective. And it also gives an opportunity to do some genetic testing. And again, this is all confidential information that is being utilized for us to better understand the progression of the disease. But it can be done without actually going to a clinic and in doing invasive medical procedures. Specifically on the bottom of the slide, you can see what we've been doing with regards to the role of GBA. And I know I want to save time also for questions and answers. So we've already looked at approximately and currently still looking at close to 250 GBA carriers from 6 countries and from 6 different countries, both having Parkinson's and also non-manifesting carriers within this effort. We're aiming to expand this. We're aiming to add an additional 250 to 300 GBA carriers. On our website right now, we're moving into a transition into the second phase. So it might appear that we're not recruiting right now, but soon we will be. So I would predict that in October to visit our site, and that we are expanding this, and we welcome people's participation, because I think the only way for us to get to a cure is for everybody to be working together and for patient involvement. Thank you so much.
Thank you so much, Dr. Baptista. And now we will have some questions. Margaret.
Hi, before we go, yeah, can you hear me? Yeah?
Yes.
Okay, before we go over to the question and answer on the screen, I received some questions when we sent out the invitation, so I'm not able to show them on the screen, but I'll read them out, and if anybody doesn't understand, please stop me. I'll try to speak slowly. Regarding the REM sleep behavior disorder, when is it considered a disorder? So in other words, if a child or a person, an adult, has this occasionally, is that something as a parent we should be alarmed about? Or maybe this is for you, Per or Ari? I'm not sure.
I think Professor Svenningsson should.
Yeah, I can try to answer. Actually, the way that we diagnose it is based on a polysomnography at the sleep clinics. So it's a quite advanced diagnosis. And then I guess if you have it, I mean, once a month or something like that, that you live out your dreams and so on, I don't think you should be worried. But if it is something where you really, you wake up your partner, or you fell out of bed and you do that once a week or something like that, you should go to a sleep neurologist, or then if there is a sleep clinics, to get this EEG measure of the brain, and which is called the polysomnography. And then one can detect that you have these specific signs that gives you the RBD diagnosis.
Okay, so it's not something that if it happens once or twice over somebody's lifetime, I mean, it's not something to panic about.
No. Actually, so it's something where people are quite handicapped about it, and so it happens at least every week, and at the moment we do have some treatments that we can offer, but they are quite it's benzodiazepines, so they are quite, it's compounds that has some abuse potential, and quite it's not therapies that we give at the first hand. But we do, if there is advanced RBD, we do have also some therapies that we offer.
Yeah, okay, thank you. And on the same theme, constipation. Obviously, you know, if somebody's constipated occasionally, it's not a problem. If you could say it was chronic constipation, or as a parent, but it could be as an adult also, should we start worrying? You know, this is a patient organization, so I'm trying to imagine that you start getting worried because your child is not performing as, you know, anyway.
I think again, it's many more people that are constipated than have Parkinson's disease. So I think the epidemiology tells us that there's twice a risk to develop Parkinson if you have constipation. It's much smaller risk than if you have this RBD, when it's a 20-fold increased risk. So again, I mean, I think if it is a child, I think the likelihood, even if we know that GBA PD may start a little bit earlier, I wouldn't be worried. And I don't think if you are constipated only, so to say, that you need to really be worried that you are developing Parkinson's disease. I think there must come some other loss of smell or some. What often happens is that if you have this tremor dominant Parkinson's disease, that the tremor comes very, very early. So you have some tremor. It can be this, for example, that you don't swing your arms properly. If you start to have some movement, then I would start to think of Parkinson's disease. But not, perhaps someone else wants to add here, but I think we shouldn't be, both for among those prodromal symptoms, I would say that the RBD is the most specific one.
Right. Okay.
I just wanted to add that I think the question really gets at the lack of good biomarkers that we have in the field, and the lack of prediction that we can do, and that's why there is such an effort and such a large amount of investment in understanding what is the sort of the signature that could predict the disease, and right now we don't have that.
Okay. But if I just take that example of constipation, for instance, and it happens, as I say, in a chronic way, doesn't that kind of imply that that Parkinson's could exist from very young age?
Yes, but yeah.
I mean, I'm saying that, and maybe something else. And
no.
I don't know, loss of smell or something, but
yeah, smell and so on. Usually, when I interview my patients, and often we then get this type of information in retrospective, then they usually say it can have happened perhaps 5-7 years before they started to have the tremor or a movement disorder. But
yeah, but I'm talking about maybe very young. Is that a possibility that it could happen when someone is?
Yeah, again, it's a little bit like what Dr. Baptista said, that we don't really know.
Yeah.
But okay. I don't. I think it's that it will start already as a child, and that you then have that for 20-30 years and then develop Parkinson. That's not really the type of information that I have received during my interviews. I think it's more like 5-7 years, perhaps.
Okay, thank you. The next question is regarding Parkinson's medicine, and it has quite a lot of potential side effects. And so for somebody who has Parkinson's disease and Gaucher disease, is this not a potential interaction between both groups of medicine? It's probably for Ari, I guess. I don't know.
Well, usually tell the patients that there are not too much overlap between or interference between enzyme replacement therapy and all the anti-Parkinson drugs. With eliglustat, it's a bit more complex because there is an issue of drug-drug interaction, and each anti-Parkinsonian medication must be checked carefully to make sure that it does not have a conflict like that. Also, some patients with Parkinson suffer from depression, and many of the antidepressive drugs are not suitable for individuals who take eliglustat, but on the other hand, all the other Gaucher treatments do not interact with the Parkinsonian therapies.
Okay, thank you. Thank you. I also received 3 questions last night, and I think you've answered them for the most part, but I'd prefer to just check. Why have carriers more possibility to suffer Parkinson than Gaucher patients? I guess it's the why is the question. Why do carriers have more possibility of suffering Parkinson than Gaucher patients?
Well, may I take it? Sorry. This is exactly the point I tried to make about the paradox, the difference between the prevalence of Parkinson in patients with Gaucher disease, who, for example, have 2 mutations, one good, one bad, and the carrier of a bad mutation has a much higher risk than the patient. My personal belief, unfortunately, I've not been very successfully convincing my colleague, but maybe I hear my 2 co-speakers will take it from here on and help me. I see only one explanation. The only explanation is that the Gaucher cells excretes a neuroprotective factor. Now, when you treat the patients, if you go to the papers of Pram history, and in the United States, all the patients receive high-dose therapy, after 10 years of high-dose therapy, this discrepancy disappears, and you see a growing number of the patients with severe mutations also suffering Parkinson. So from my perspective, this is the answer. And another claim for what I talked a lot about, the core problem of misfolding protein problem, gets support by a few recent publications that showing a high prevalence. I mean, Professor Svenningsson presented one of them. There are some mutations or changes, nucleotide substitutions in the glucocerebrosidase gene that are a risk for Parkinson, but they are not disease-causing allele, like a polymorphism or something. So they're like genetic markers. So if it doesn't cause any disease, it does not lead to any storage. So I think at the end of the day, it will be a combination of several things, but for the question of the mutation is easier to answer than to the other questions like Dr. Baptista's talk about why many patients, the majority, do not develop Parkinson. So there is a lot to be studied yet.
Yeah, I understand that. Then the other question is, if the Gaucher patient started with the disease when she was 4 years old, has she got more possibilities of suffering Parkinson than starting later, than with a later?
Well, it goes again to the issue of severity.
Yeah.
The younger the age at presentation, or the more severe the Gaucher is, and if a patient with severe Gaucher will be treated for many more years with enzyme therapy, if the patient belongs to that group that had a higher risk for Parkinson, probably the risk will be greater. But we are not there yet to answer.
Okay.
Unfortunately, too many unanswered questions.
Yeah, but at least the questions are being asked, so, you know. And the last one is, my daughter was Gaucher type 1 and my father suffered from Parkinson when he was 50 years old. Is there a connection?
Well, the connection is probably by the higher prevalence of Parkinson in relatives of Gaucher, and most of these relatives are carriers. So we go back to the fact that you are a patient, so your parents and grandparents and uncles and all the relatives are at high risk to be a carrier. I can tell you that from our data, more than 25% of the patients will give a family history of somebody in the family that has Parkinson. We talked to Roy Alcalay. He usually doesn't trust family trees beyond the generation of parents, but I think it's pretty impressive.
Okay. Okay. So I'll just go over to the question response. Can everybody see that? What's showing or not? No? Nobody can see it. Well, then I'll read it out, okay? Is there a risk, is the increased risk with all mutations for Gaucher disease or only certain mutations? Well, I think you answered that one already. Yeah. So, okay. Wilson, as a patient with Gaucher disease, if we have some ascendants in family having Parkinson disease, is there a possibility that this ancestor might be the carrier of the GB1 gene?
Yes.
Yes. Okay. Which are the exams to perform in preclinical PD to confirm that PD will develop?
That's a very, this is what Dr. Baptista is really trying to find out. So
yeah. Okay. And then what is the diagnostic test to diagnose Parkinson?
it is a clinical examination still. So it is that they should have, if you have a Parkinson diagnosis, you should have the slowness of movement or the bradykinesia. That's, you need to have that, and then you should have rigidity, and in many cases then also tremor. But tremor is, there is a form of Parkinson where you don't have tremor.
Well,
yeah, diagnosis appears to be a very tricky thing to do, especially if you might have another type of synucleinopathy, such as MSA, and trying to differentiate it. But then again, responsiveness to levodopa can sometimes clarify that.
That's true. We often, there are what we call red flags that we need to look for in the beginning. If it can be this, we call it atypical forms of Parkinson. I mean, it's about 10%, 10-50% that have those forms. And exactly like Dr. Baptista said, they respond much worse to the treatment.
I think we have time for one. There is the one from the chat. Patients in some countries have some tests built into their GD assessment for multiple myeloma. Should we be looking at the same approach for PD?
I wasn't sure that I understood the question. For multiple myeloma, there are some laboratory tests looking at monoclonal gammopathies, and there is not a specific test that predict Parkinson, as we have heard. At the moment, no.
Yeah, okay. Okay, thank you very much to all participants and to all panelists. And before we, before we close this webinar, I just would like to share one slide. We have my colleagues from International Gaucher Alliance prepared more webinars for September and October, and more details you can get at the website. I think that's it. Thank you very much for participating, and especially to our panelists for giving us these great lectures. And Margaret, thank you very much for your help.
Thank you very much for the invitation.
Thank you.
Thank you.
Yeah. Goodbye, all.
Bye.
Bye-bye.
Bye.